Hello everyone, and welcome to today's webinar. You have joined the presentation using your computer speaker system by default. However, the audio is usually more reliable via phone. If you would prefer to join over the telephone, just select phone in the audio pane and the dial-in information will be displayed. You will have the opportunity to submit text questions to today's presenters by typing your questions into the questions pane on the control panel. You may send in your questions at any time during the discussion. We will collect these and address them. If your question is for a specific panelist, please include his or her name in your question. Today's webinar is being recorded and you will receive a follow-up email within 48 hours with a link to review the recording. I would now like to welcome Dr. Kara Bortone, Senior Director, Portfolio Sourcing and Management, to kick off today's program.
Welcome everyone, and thank you for joining us. Before we get started, I'd like to provide some brief background on Johnson & Johnson Innovation. Next slide, please. J&J's Chief Scientific Officer, Dr. Paul Stoffels, is at the core of Johnson & Johnson's innovation model. Innovation is occurring all over the world, but how do we harness it to bring solutions to patients? How do we work with entrepreneurs and academics and enable them to take their products and technologies from concept to commercialization? Johnson & Johnson Innovation was founded to provide a solution for this. On the next slide, you'll see our co-creation model. At Johnson & Johnson Innovation, our aim is to advance innovative products and solutions to improve human health by partnering with and enabling innovators to advance their technologies through access to a range of incubation and commercialization resources. Our focus is on three main sectors: consumer health products, medical devices, and pharmaceuticals, as well as cross-sector solutions. On the next 4 pillars slide, you'll see that we provide a comprehensive global solution to support innovation through 4 pillars. Our J-Labs incubation facilities provide early-stage innovators with state-of-the-art infrastructure and access to J&J commercialization expertise. Our innovation centers accelerate innovation through creating collaborations with innovators and around the world from inception to early-stage development. And JJDC is Johnson & Johnson's strategic venture capital arm, a long-term investment partner to healthcare entrepreneurs. And lastly, our business development arm supports long and valuable relationships through licensing, M&A, and alliance management. As you can see on the next slide, a map of the world, Johnson & Johnson Innovation spans the globe and has presence in major life science hotspots worldwide, including 13 J-Labs facilities and 4 innovation centers. In the final slide that we'll talk through today, you can see the J-Labs movement slide. J-Labs innovators provide a community for early-stage life science innovators, equipping them with access to resources, commercialization, expertise, network, infrastructure, and more, enabling them to accelerate their healthcare solutions. If you're interested in joining J-Labs, please visit the J-Labs website to submit an application or contact the event organizer with any questions. And now we'd like to move to our panelists. In this fireside chat, vaccine expert Dr. Jason DeHart, Senior Principal Scientist at Janssen R&D, will guide a discussion with Dr. John Lewis, founder and CEO of Entos Pharmaceuticals, around the current DNA vaccine landscape in infectious diseases and how Entos' proprietary platform for genetic medicine delivery may enable the formulation of a potential DNA vaccine for COVID-19. Without further delay, let's welcome John and Jason.
Hello, hi, Kara.
Hey, John, how are you doing today?
I'm doing not too bad, Jason. It's nice to see you.
Excellent. It's good to see you as well. So thank you for taking the time to talk with us today about the work you and Entos are doing to develop a DNA-based vaccine against the pandemic coronavirus SARS-CoV-2. You know, I'm sure everyone on the line is acutely aware of the tremendous amount of effort that's being put in by many groups across the world to develop a vaccine to protect against COVID-19. You know, I'm excited to hear more about the work that you and Entos are conducting on this front. But I thought, you know, as we get started, it'd be great if you could tell us maybe why and how Entos decided to focus on a vaccine for SARS-CoV-2.
Yeah, no, absolutely. It's a great question. Yeah, well, thanks so much, Jason. So Entos Pharmaceuticals is a Canadian company with a platform technology to develop and deliver genetic medicines. So Entos was founded in 2016. We're a Canadian company with a site in Edmonton, Alberta, research labs and offices up there. We're also, really proud to be a resident in JLab San Diego and have labs and offices in San Diego as well. And so our Fusogenix platform, we've been developing with scientific co-founder Roy Duncan, another Canadian scientist, for about 15 years. And the purpose of this technology is really to join sort of the lipid nanoparticle field with the viral delivery field in a formulation that harnesses sort of the virus's ability to deliver DNA and then lipid nanoparticles benefits for scaling up and manufacturing. And so we were in Entos developing this platform technology for use mainly in oncology purposes. And we were planning to start our first in-human clinical trial for an oncology solid tumor application early next year. So of course, the world has turned upside down in that time. We were reading all the news in interest in December. As January and early February came along, it was apparent that this was going to be a worldwide pandemic. And we sort of looked at sort of the solutions that were coming out for COVID-19 vaccine and potential immunotherapeutics, and we thought, there are lots of different platforms available. We thought, the Fusogenix platform was in a particularly good position to deliver a DNA vaccine, and we like the idea of a DNA vaccine for several reasons. DNA is it's very stable. We know the manufacturing and scale up very well, and it brings a lot of flexibility in sort of the kinds of antigens that you can insert for SARS-CoV-2. So we sort of had a all hands on deck meeting in early February. Looked at the technology, looked at the opportunity to make an impact, and decided to go full speed ahead with the COVID-19 vaccine.
Excellent. I think that's very informative, and I think as we go through the chat, it'll be interesting to dive into a little more detail what goes into the process of developing a DNA-based vaccine. But before we do that, I thought it might be nice, particularly maybe for those on the line who may not have a deep background in vaccine development, if you could give us, maybe a high-level overview of some of the critical steps that go into developing an effective vaccine.
Yeah, absolutely. Well, I mean, as many people are well aware, humans are susceptible to a wide variety of different pathogens in our environment, be they bacteria or viruses. Viruses in particular are can be very dangerous, and certainly when they emerge very quickly, and we don't have an inbuilt immunity, they can have a tremendous negative impact. So I mean, normally our immune system does a pretty good job of recognizing foreign things that enter your body. Viruses in particular are interesting because they need your body to sort of survive and multiply. So they'll maybe gain entry through the nose or the eyes or the mouth, and they need to get inside your cells to be able to replicate and make many more copies of themselves. So at this point, your innate immune system, will particularly recognize them and mount sort of a first attack, but it's really the adaptive immune system, the ability of your cells then to specifically recognize this new foreign pathogen and develop a cell-based response, like a T-cell response, and then an antibody-based response that can sort of soak up the virus before it can infect. So generally, if we get infected with a virus, there's this arms race that goes on, speeding to clear the virus before we get really sick. And but once your immune system beats the virus, you retain the memory for long term. So you can produce antibodies and a cell-based response really quickly after getting infected a second time. So vaccines sort of shortcut this process. So with the vaccine, what we want to do is we want to introduce pieces or a non-virulent form of the in this case SARS-CoV-2, so that your immune system gets a chance to look at it, to produce this adaptive response, whether it be through antibodies or cell-based response or both. And then when we actually get exposed to the SARS-CoV-2 virus, we can mount an effective response and be protected from the virus. So from a vaccine standpoint, there's a few different things we need to think about. We need to think about sort of what the vaccine platform we're going to use. So traditional vaccines, probably the majority of the ones currently in use, use pieces of the virus. So either it's the whole virus inactivated, so it can't affect you, but your body can see all the proteins, or what's called subunit vaccines, where certain proteins are produced and injected so your body can see it. These traditional vaccine approaches have produced very effective vaccines against a number of pathogens, but the time to development can be very long. I think the average discovery to approval of a vaccine is over 10 years. So in a rapidly emerging pandemic situation, it's just not feasible, and it's certainly challenging to be able to take a traditional approach. So now, with a lot of innovation in this space, we have the opportunity to develop genetic vaccines. So these are vaccines that use our tools of our own genetic code to insert the instructions for making the SARS coronavirus proteins in our own cells, and these can take the form of RNA vaccines or DNA vaccines, and then really the challenge becomes, how can we formulate and deliver this DNA and RNA instructions into the body? And there's a number of ways we can do this. We can do this by taking a different virus. So viruses are really good at injecting DNA. So vaccine platforms like the adenovirus platform, so the Oxford group and others, I think, and J&J is working on adenovirus-based platforms. So adenoviruses do a great job of delivering actually DNA payload into cells and making the SARS-CoV-2 antigens. And then there are non-viral platforms, so lipid nanoparticles, so basically taking a mix of lipids that are generally already present in your body and putting the DNA or RNA inside that and injecting it. And companies like Moderna and CureVac and BioNTech are using a messenger RNA lipid nanoparticle approach. And then Entos' approach is slightly different. I mean, like I said, the key part of our Fusogenix technology is that we manufacture on a lipid-based platform, but we deliver the DNA using a viral originated fusion protein that we've optimized specifically for DNA delivery. And this is really important because when we're looking at RNA and DNA, we think there are a few compelling reasons to choose DNA, if we can overcome the challenges of creating a drug that can, express those antigens in a person and provide protection.
Excellent. Thank you for that overview. And so, I guess, for me, a takeaway from that is you need antigen selection is critical, your platform is critical, and then your delivery and formulation is another critical component for development of an effective vaccine. And then, of course, you got to think about scale up and distribution. So, if we start to think about antigen selection, what was the approach that you took at Entos to identify a CoV-2 antigen, and maybe why did you decide on the specific antigen that you've included in your DNA-based vaccine?
Yeah, absolutely. No, the antigen selection is key to producing a robust immune response. We took the position that having a DNA or a genetic-based vaccine, we're able to, so the chemistry of DNA is the same for every vaccine, except we're able to encode different epitopes or even mixes of different epitopes in a single platform. So we sort of looked at the emerging science that was coming out in December, January, early February, and looking at our experience, globally from other coronaviruses like SARS and MERS. There were some ideas that we could use. So first of all, with the SARS epidemic a few years ago, there were no effective vaccines produced, and there were several reasons for this. One of these reasons was the induction of what's called antibody-dependent enhancement. So this idea that you can produce antibodies against a pathogen like a virus, but some of these antibodies in coronaviruses actually facilitate or help the entry of the virus. So it was key to produce a potent neutralizing antibody response to prevent infection without creating this antibody-dependent enhancement. So we created a few partnerships, created a key partnership with a company called Epivax in Rhode Island. They have 20 years of experience looking at epitopes, deciding which ones can promote or reduce immune responses. And so the goal for our DNA vaccine was, because we were able to rapidly prototype several different mixes of antigens, our goal was to produce a potent neutralizing antibody response, but also, and I think perhaps more importantly, produce a potent cell-based, so a T-cell-based response, and in particular, for immunologists out there, either a balanced T helper 1, T helper 2 response or a T helper 1 bias response. We saw in some of the cases of the SARS, experimental SARS vaccines, they promoted a Th2-based response, which can create a lot of various symptoms that are similar to the acute respiratory symptoms we see in severe COVID-19 patients. So we've been spending the last 2 months, we call it rapid prototyping, but testing mixes of antigens that promote that balanced response between antibodies and cell-based response. In the end, so we've just selected our lead candidate just this past week. It can, like many other companies, it incorporates key components of the coronavirus spike protein. This is the protein that coronavirus uses to enter cells through the ACE2 receptor. And so we already know from other people's work and early human clinical trials that can promote a strong immune response. So we think this is key to provide protection. We also have other epitopes in there, cell-based epitopes. And the goal there is to potentially provide protection not only against SARS-CoV-2, but against antigens that are conserved throughout other coronaviruses. So the hope is, and we haven't tested this yet, that it'll provide some measure of pan-coronavirus protection.
Okay, excellent. That's very exciting. So I think a follow-up to that is, and I think you touched on this a bit, right? Understanding the type and magnitude of immune response the vaccine needs to elicit is pretty important. For some viruses, a neutralizing antibody response is critical, and we understand the titers, the level of neutralizing antibody titers that need to be induced to provide protection, for example, in influenza. Are there any known correlates of protection that have been identified for CoV-2? And, it sounds like you're trying to take a balanced approach to get a good T and B cell response here. But if we don't understand the correlates of protection, what risk does this present to vaccine development?
It's a great question, and it's something that all vaccine developers are struggling with, this idea that we need to commit to a sort of a lead candidate without having the full information. So there's a lot we don't understand about SARS-CoV-2. You mentioned correlates of protection. I think it's, so we don't have any great correlates of protection right now, although it's emerging. So certainly data from other labs and ourselves indicates that the overall level of for instance, anti-spike protein antibodies produced in an individual correlates really well with its neutralization ability. So I think from the vaccine standpoint, we have a relatively good idea. If we can promote more antibody production, we can promote a more robust neutralizing antibody titer. What we don't have a good handle on is sort of in the human population, what level of neutralizing antibody titer is sufficient to fight off the virus, or is it that the neutralizing antibody titer in particular that's critical for that? So certainly in our work, we've analyzed quite a few convalescent patients, and there's been a lot of published data on this. And there's a huge range of antibody titers, several orders of magnitude difference. And some of these lower neutralization titers seem to be sufficient to protect from the virus. So it's curious why some individuals have very high levels. And then there's the other sort of emerging evidence around durability of the response. So we're seeing in multiple studies, including one published yesterday, that over time, that neutralizing titers in people who have had SARS-CoV-2 are decreasing over time. It's not completely clear yet, so we can draw some conclusions, but I think a little bit premature for that. I think obviously the cell-based component is critical, and whether these convalescent patients can mount a good immune response, but that'll be something we have to look at. What is the durability of these vaccines over time? Because it's perfectly reasonable to think that the first vaccine approved through the gate provides sort of an acute, good short-term protection. We may need other solutions down the road for longer-term protection. Excellent,
thank you for that. Let's talk a little bit more about the platform, the DNA, the plasmid-based platform that you're advancing. You talked a little bit about some of the advantages of using DNA. Could you maybe expand on that a little bit more, and maybe you could talk about the size of payload that you're able to encode and number of different antigens?
Yeah, absolutely. Yeah, as I said, for genetic vaccines, whether they be RNA or DNA based, we're delivering the instructions to make the coronavirus proteins. So DNA was sort of first shown. You can inject sort of naked DNA into a muscle and express proteins. That was shown late 80s, early 90s. In 1993, it was shown they could inject a naked plasmid DNA encoding influenza proteins, and you're able to get protection against multiple strains of influenza. So the idea that we can produce a vaccine from DNA has been around for quite a while now. The real challenge has been in actually formulating something that works in people. And the key challenge is that delivery. So while you can inject large volumes of DNA into a muscle and get some expression, to create this reproducibly in a vaccine without a dedicated delivery system has shown to be challenging, if not impossible. And there's currently no licensed genetic vaccines available, although there are DNA vaccines available licensed for animals. So I think the challenge here is really in the delivery, and this is what our platform is designed specifically to address. So to provide a little more color on what the platform is, so we're from a manufacturing standpoint, we're a lipid nanoparticle. So we incorporate lipids that normally make up the outside membrane of our cells that are well characterized and well tolerated. We have a lot of clinical evidence to show that they're going to be well tolerated. And then mixed in with this is this fusion-associated small transmembrane protein. I'll spend a little bit of time talking about it because it's really cool. So Roy Duncan, a Canadian virologist on the east coast of Dalhousie University, about 16 years ago, was playing around with an orthoreovirus that infected alligators and birds. And what he found in a dish is that when you infect cells, they all rapidly fuse together. And so, as any scientist who's like, wow, there's a new fusion protein here, and he went and spent the next few years trying to characterize it. And it actually took a lot longer than he expected because that protein was so small, and you'll appreciate this as a scientist, that it was so small that it ran off his gel. So it was so small, he assumed it was larger like the other fusion proteins. And it took him, maybe 2 or 3 years to see that it was really, really tiny. Smallest known fusogenic protein by several orders of magnitude. Really, really tiny ectodomain, which is important because then it's non-immunogenic, so you don't get an immune response against the protein itself. But what's critical is that these proteins were sufficient to induce membrane-to-membrane fusion. And so Roy went on in his academic lab to characterize these fusion proteins, very similar ones from different orthoreoviruses. And we started working together about 12 years ago, basically doing structure function analysis of the different fusion proteins and sort of mixing and matching the components to come up with a fully synthetic fusion-associated small transmembrane protein that we now incorporate into Fusogenix. And what's great about this, when we manufacture sort of these 3 lipids together with this fusion protein, encapsulating DNA, it allows that nanoparticle now to fuse directly with the plasma membrane of the cell. So unlike other DNA and RNA delivery platforms that rely on endocytosis and then escape from the endosome, we deliver directly through the plasma membrane. So for instance, if we inhibit endocytosis, it still does a great job of delivering. And the plasmid DNA platform that we're using, so I guess one of the main differences between messenger RNA and plasmid DNA is the plasmid DNA has to get to the nucleus to produce its proteins. Messenger RNA can go to the cytoplasm. We're working with a great plasmid backbone, getting into your question about size, that's very minimal, less than 1,000 nucleotides form the whole backbone. It does a great job of getting directly into the nucleus. When Fusogenix delivers it to the cytoplasm, we get very robust expression. So we're looking at in the mix of, so the backbone's very tiny, so that allows us to incorporate, actually have a lot of flexibility in the amount of cargo we can put in. With our platform, we make it with microfluidics, so they're self-assembled particles that are formed in a very controlled stream. So as they're self-assembled, we can put in plasmids that are huge, so 15, 20 kb. They'll self-assemble into particles that we can then use. Obviously, as they get larger, their ability to inject DNA decreases somewhat, but there's really no size limitation. So we've had the flexibility to look at a large mix of both single antigens and multiple antigens.
Okay, excellent. Thank you for that explanation. So following up on that then, you mentioned how DNA, right, needs to get into the nucleus in order to get expression of your antigen, where your mRNA does not. You just need to deliver that to the cytoplasm. Does your Fusogenix platform then work for mRNA, and does it potentially work better in an mRNA space where you just need to get it into the cytoplasm as opposed to maybe a DNA-based vaccine?
Yeah, absolutely, yeah. So we've optimized, because the fusion protein is doing sort of the heavy lifting for delivery, we've optimized formulations to encapsulate, RNAi, siRNA type constructs with tiny nucleic acids, messenger RNA, plasmid DNA, and even for gene editing. So, Cas9 gene editing proteins with the guide RNA. So there's really no limit to the kinds of things you can encapsulate and deliver. When we looked at the landscape, though, there are companies already well established and ahead of us in the space working on messenger RNA. And while messenger RNA is really compelling from the standpoint that we can rapidly prototype and test a vaccine, we've seen that. It's really good. It has a very potent high expression in the short term, which I think is essential to produce a good immune response in a vaccine. But messenger RNA, sort of by its biology, has challenges in the fact that it's designed in our bodies to be transient. So it's quite unstable, and so that presents challenges. So there's been chemistry figured out to make it more stable, which is great. But generally, thinking about the scale up and production of a messenger RNA vaccine, we're looking at potentially having to keep them, very cold, so -80°C, from what I understand, and then transferring a vaccine, to billions of people around the world. Certainly in first world countries, it might be straightforward to do that, but I think it's going to be a real challenge to give it everywhere. So I think, and I think many people have said this before, I think it's going to take more than one delivery platform to be able to solve this crisis. And I think DNA is particularly well suited because of its stability. So for instance, our formulation, as we currently have it, is a suspension, liquid suspension that's injected intramuscularly. Our stability studies indicate that it's stable in that formulation, so in liquid form, for over a year. So this makes it amenable to delivery around the world. We're also investigating the idea. So we have some really exciting data showing that if we deliver it orally in a pill form, that we get great expression of a plasmid transgene. So we're investigating this, and again, this is we'll wait and see if it works well or not for a vaccine to see if we can produce a similar immune response through an oral route. And I think that'll be a real game changer as far as being able to make this and distribute it in areas where cold chain just isn't available.
Interesting. That's I think that's a good point. So if I understand correctly, then it's stable at room temp in solution, or so you've eliminated essentially the need for maintaining the cold chain.
No. So we're currently in suspension. It's refrigerated, so 4°C, and we have stability data out past a year for that. For the oral form, we can lyophilize it or dry it down, and then we can either reconstitute it later and inject it, or just ingest it as a pill form. And again, that's something we need to figure out and test and develop. But certainly everything we've done so far indicates that that's a possibility.
Okay, excellent. Let's talk about scale up and manufacturing a bit and distribution, right? I think that is a challenge that everybody is going to be facing, that are working to develop these vaccines to protect against COVID-19. I think the last count, there's somewhere around 130, different groups in that range that are really pushing forward to do this. As everybody is moving towards manufacturing and trying to do it quickly and do it simultaneously, I guess what issues do you think everybody is going to be facing as they move into the manufacturing phase?
Yeah, that's a great question. I mean, the pharmaceutical industry is deeply entrenched in planning and timelines, and often, for developing an oncology drug, for instance, you're planning out a timeline that's 7 or 8 years in the future. So that gives you a lot of leeway to think about how you're going to scale up manufacturing, to make investments at the proper time to be able to build that manufacturing capacity and expertise, and at the same time engage with regulatory authorities to bring, as you manufacture different scales and qualities, to bring that through regulatory approval through phase 1, phase 2, and phase 3 trials. Well, all bets are off with COVID-19. We need to, every day that we don't have a vaccine, carries a significant cost, both in health-wise and economically. So I think everybody certainly agrees that we got to move as fast as we can. And so this has been a real challenge, because now we need to think about things like, well, number one, preparing for clinical trials as quickly as possible. Obviously clinical trials, especially the early part, the key is safety. And for vaccines, this is extremely important, because we're vaccinating a healthy population. So the the bar for any side effects is very, very low, or sorry, very, very high. And so, but then we need to think about, so if we do have a vaccine, we bring it through phase 1 and phase 2 clinical trials, we show that it's safe, we show that it produces a very strong immune response. Okay, so now we potentially have a solution on our hands. How are we going to deliver this to, to 6-7 billion people in the shortest amount of time? And this is a monumental challenge. So, we're contemplating and being able to build commercial manufacturing facilities before we know the vaccine works. And you've seen this in the news. And many companies have invested heavily in building out that commercial manufacturing capacity far ahead of the time when they're going to know that their vaccine is an effective countermeasure against SARS-CoV-2, and we're no different. So for our manufacturing platform, we really like, the scalable microfluidics platform we're using is extremely scalable, and we can sort of expand it in parallel. So just imagine we have these microfluidic box. We flow two things in one side, so basically the DNA and the lipids and the fusion protein mixed together under these controlled circumstances and produce the fully formed vaccine at the other end. We have a single purification step after that, and then we put it into vials. So that sounds really easy. So I guess the bulk product part of that is pretty straightforward. So we can make, with a single microfluidic platform, we can make roughly 1 million doses a day. So you can make about 100 liters in a single run and run that every day. So I think for us, that's really compelling as far as scale up. We can put this in different countries and scale up manufacturing very quickly. I think the challenge that we're facing and everyone else is facing is this last step. It's called fill finish. So actually taking now that bulk product, putting it into individual vials, labeling them, ensuring their sterility, et cetera, and for a cancer drug where you're making maybe a couple thousand doses, it's pretty straightforward. If you're talking about now making 200 million doses, it's a staggering challenge. And pair that with the fact that we have dozens of companies sort of moving their vaccines forward, reserving fill finish capacity, hoping that their vaccine is going to be effective. I think we have a huge challenge. And so and certainly it's a challenge for us to think about how we're going to do that fill finish.
Yeah, I think that's a great point. And as you're talking about that, I wonder, with so many people, so many groups trying to push a vaccine forward, I think clearly we'll need more than one vaccine, right? As you mentioned. But does that put additional strain on that supply chain, on that fill finish? And does a point come when maybe everybody needs to get together and collectively just decide, okay, maybe we need to advance maybe a handful of vaccines to that point?
No, that's a great point. I think attrition is going to happen. But again, I mean, we're at this stage of the game. There are many very promising candidates, and I don't think we can make those tough decisions yet without sufficient clinical data. And I think as time moves on, that clinical data will come. And there's the funding aspect. So institutional and government bodies are stepping in, and private foundations are stepping in to invest in manufacturing capability ahead of time. Bill Gates has talked about simultaneously funding 7 different manufacturing processes, and I think this is important and critical if we're going to, once we have a winner or even a series of, it could be conceivable that there are 4 different platforms that are viable and can be produced commercially. So I think it's a good idea to do this, and I think we're going to have to, as these vaccines. And I'll talk about coordination. So the WHO is obviously involved in mediating these conversations between different groups. The Entos team, there are 5 of us involved in different levels of the WHO. Their committees on assays and animal models and clinical trial evaluation. And I think these conversations are really important. First, to coordinate the scale of phase 3 trials. So just to back up a step, first is phase 1. Phase 1 is really safety. We can get some indication of how well it works or stimulates an immune response, but really is to determine its primary safety. Phase 2, you expand a bit and you look even more at the endpoints for immune response. But phase 3, and particularly for a vaccine, is very challenging because we need to test in tens of thousands of people to establish safety and protection. And if you have 80 different vaccines competing for space and time and resources, it's gonna be very hard unless it's coordinated. And I think so. The WHO is doing its part to try to coordinate, vaccine testing and approval. It looks like there'll be some worldwide phase 3 trials. We're looking if it makes sense to be a part of those as well. And then, a little more contentious issue, I may as well bring it up here, is the idea of a challenge study. So Entos has been sort of engaged with regulators, mainly in Canada, and this idea of, can we take healthy individuals who have a relatively low risk of having serious consequences of SARS-CoV-2 infection? Can we vaccinate these individuals with an experimental vaccine and test to see if we're being protective? And I think certainly, and actually somewhat to my surprise, the regulators have been open to this idea. I think it's an evolving international conversation, but it may be one way to speed up sort of deciding which vaccine makes sense to move forward.
Excellent. Yeah, so following that up again, multiple different vaccines being developed. I would imagine a lot of them are a similar approach in terms of encoding the spike protein. Some may be slightly different in terms of, encoding spike or other antigens to try and elicit a T and B cell response, a balanced response there. So they may perform differently. We know, particularly in older patients, they tend to be a little more susceptible to infections, for example, from influenza, and so therefore require special vaccine formulations or different dosing for the vaccine to be able to confer protection. When you think about that in the case of a CoV-2 vaccine, how do you think the vaccines that are being developed may be able to protect against different populations, whether it's the older individuals or populations who just may be more susceptible?
Yeah, no, that's a great question. So, I mean, we're looking at what's happening to our populations as SARS-CoV-2 sort of moves its way around the country, and it's very obvious that there are more vulnerable populations than others. And I think from a vaccination standpoint, we want to build a herd immunity, and this could be done in protect the older population through vaccinating all the young, healthy adults. And I think that's one way that we can protect the elderly population. But the other is, will a vaccine work in elderly individuals? Certainly, as you mentioned, our experience with the influenza vaccine, they require, potentially much higher doses to elicit the same neutralizing antibody response. And so certainly at Entos, we've been thinking about and designing our phase 1 trial to incorporate different dose levels and tranching in different age populations to incorporate this. One of the things I'll mention too is the design of trials is really critical to the speed that we can move forward. So looking at the sort of adaptive design where we can start with a phase 1 in a young healthy population, as soon as we establish safety, move that into phase 2, but then look at then safety and with a higher dose with older individuals, and then move that into phase 2 when we establish an effective dose. So I think there's a lot of things at play that need to be incorporated. One of the things, it's probably useful to mention this at this time, there's been a lot of talk with SARS-CoV-2 about neutralizing antibodies and specifically identifying an excellent neutralizing antibody and then producing that antibody as a drug itself. So the idea, and the same idea for taking convalescent patient plasma and infusing it in patients currently suffering from COVID-19. So producing these antibodies. So I would say that we're really interested in utilizing the Fusogenix platform in addition to a vaccine, but also looking at production of therapeutic antibodies. So we've created some partnerships, and we're looking at we're just in the process of securing funding to launch a therapeutic antibody program. So I think for me, and thinking about my parents and other individuals who are more susceptible to advanced or to severe disease, is I think this neutralizing antibody therapeutic approach may be a solution to address the elderly population in places where a vaccine just might not work as well.
Excellent. So when thinking about clinical trial design and who to include or not include, as you're looking at, for the development of your vaccine, do you anticipate including any sort of serological antibody testing in your trials to screen out maybe those who already have been infected and have a response? And how are you looking at that?
Yeah, no, that's a great question. So I would say, so absolutely. So I think previous exposure to the virus is a key part of looking at the immunogenic response to a vaccine. I think it's much more important for the phase 2 portion than the phase 1. You know, we're looking at here at Entos and many other companies a balance of speed and data, right? So I think it's critical for, you know, for us particularly, our Fusogenix platform hasn't been in humans before. Again, we were planning a first-in-human oncology trial early next year. So this, you know, we're looking to start our phase 1 trial in August 2020, and we're thinking about, you know, we want to establish the safety of the platform and also our specific vaccine candidate. As we move into the phase 2 portion, it'll be really critical for us to look at the immune profile of that patient. Have they been exposed to SARS-CoV-2, and how does that affect their ability to be vaccinated?
Excellent. So we got a few minutes here left, and I wanted to kind of talk about the business side of things, the business aspect, right? So vaccine development is very expensive. Some companies have partnered with government agencies on the development. Others have opted to foot the bill themselves. You know, as a smaller company, as a smaller biotech, there are quite a few of these. You know, how do you go about navigating the need for capital to fund these studies, to fund the development? And I guess as a follow-on to that, can you expect really a return on your investment for that, you know, for this work, or does that even matter for any company at this point?
Yeah, no, that's a great question. So I think as we go farther into this, people may think of that a little more than early on. Certainly early on, for us, the decision was we were either going to not do any vaccine, or we were going to go full speed and try to come up with something that was a solution. I don't think there was any in between. And I think the team is all galvanized behind the idea that we have a great solution and can get there quickly. So I think we're all in as far as a company goes. And as you mentioned, it's not cheap to take a program from scratch and take it through clinical trials and manufacturing in a short period of time. So at Entos, you know, specifically, we're a profitable company, which is not all that common for a biotech. And we've basically taken our profits and reinvested them in this program. We've been fortunate to have support from the Canadian federal government, and some of that support will fund the vaccine all the way through phase 1 clinical trials. So we've been, you know, a part of it is, so as you mentioned, some companies, smaller companies are partnering with larger companies. We're certainly open to this. For me personally, it was really important for us to build, number one, a lead candidate that we felt very confident in going forward, and number two, build the data package where we thought it had a, you know, fantastic chance to be one of the solutions that would make it to the finish line. And so as we look, so I think Entos itself, I think we can, you know, through the funding mechanisms we have in place, we can definitely get to the end of phase 1 and pretty much all the way through phase 2. Where it will be critical to have partners is sort of that next phase. Once we know in our phase 2 trial that we have a candidate that we want to, you know, deploy and commercially manufacture, it'll be key at that point to have, you know, partners to help us build that commercial manufacturing capacity and have solutions that'll be different depending on the country. So I think we have a GMP quality manufacturing plant in Canada for Entos. We can manufacture all the doses for Canada, but we're going to need to think about that manufacturing in the US and Europe and other countries.
Excellent. Thank you very much, John, you know, for all your insight and for sharing the work that you've been doing at Entos to drive you know, to push a vaccine forward, a DNA-based vaccine with your platform. I think it's very informative, and, you know, I hope that you guys are successful. I hope we have lots of success stories on this front. I think it'll only be beneficial for everybody. The sooner we can get a vaccine, the better. So at this point, I think maybe we'll turn to some of the questions that those on the line may have. Let's see here. First one I'm looking at is, they say from their understanding that the plasmid that you're using is encoding certain epitopes from the spike protein. Why not encode the entire spike protein?
Yeah, no, that's a great question. So certainly we do have candidates that encode the entire spike protein, and judging from data from our own experiments and other labs, full-length spike protein does seem to to be the most immunogenic. And so absolutely. And we're also looking at incorporating the epitopes from other conserved coronavirus proteins.
All right, excellent. And could you expand maybe a little bit more in terms of how cell-based protection in the past has correlated with a clinical response to vaccines? So you talked a little bit about, trying to induce a good T cell response, a good CD8 response, to clear the virus. Is there correlates of protection either for SARS or other viruses where you can correlate this cell-based response to a vaccine?
Yeah, so certainly I wouldn't say the data is a slam dunk, but certainly there is evidence out there, particularly in there's been a lot of talk and work around this enhancement. And so what we, from several studies in animal models, and that's important that they're animal models, show that the type of T cell response you get is important. So inducing a strong T cell response, I think, is critical to provide long-lasting and durable protection. But that T cell response must be balanced, because there's evidence in patients, in animals that developed more severe disease after being vaccinated, that this response tilted toward a Th2 response. And so these Th2 responses can be linked to things like adjuvants. They can be linked to the specific epitopes. And so I think the science is just emerging on how we evaluate this using either cell-based or animal-based assays. And so we're, as we move this forward, we're looking to the science that's developing to try to make sure we make the right decision as we move forward with the candidate.
Excellent. Yeah, and just real quick follow up on that. Does your platform require adjuvants?
Yeah, that's a great question. So if you're looking at a traditional subunit or an inactivated vaccine, there are well-established adjuvants, and now there's a lot of focus on utilizing adjuvants that promote that T helper 1 or balanced response. For genetic vaccines, it's slightly different. So we have so just to back up a bit, an adjuvant is typically something you formulate with the vaccine to purposefully stimulate a kind of response, or either jack up the immune response in general, or direct it toward a particular part of the immune system to get sort of the qualities you want in protection. And so with a DNA vaccine, we have a few different tools available. So we don't really use traditional adjuvants that we formulate with the nanoparticles, but we can encode, actually, because of the DNA and really the lack of limits on size, we can incorporate into the plasmid backbone different things that can promote the kind of immunity we're looking for. So things like CpG, CpG formulated as an oligonucleotide has been shown to stimulate TLR9 and increase the immune response. Although it's sort of it's a balance, but TLR9 can also shut down protein production. So there's a balance there. So we have actually genetically incorporated CpG RNA components into our plasmid backbone. That seems to have a beneficial response. It looks like the candidate we're going forward will have that. There's also another component built in that stimulates the RIG-I pathway, and stimulation of the RIG-I pathway is well correlated with more enhanced immune response, so being able to lower the final dose. And so it looks like our final vaccine candidate will contain both RIG-I stimulating factors and CpG.
Excellent. And so do the different fast proteins display different cell tropism? So can you target your formulation to different cell types, or does it just fuse with any cell?
Yeah, that's a great question, Jason. Spent my academic career developing targeted nanoparticles that can target specific ligands. With a genetic vaccine, though, I'm really embracing the idea of being able to deliver to every cell and then have the payload be selective for what kind of response we want to get. Because delivery has been such a challenge, I think being able to deliver a lot of DNA promiscuously actually is an important component. So I'm using that to lead up to the fact that the fusion proteins are purely biophysical. So they don't have any ligands they interact with. They don't have any receptors they interact with. It's a purely biophysical interaction between the lipid nanoparticle and the plasma membrane. And so there's really no tropism to any cells. Now, that said, of course, I have an academic lab that develops targeted nanoparticles. We can engineer targeting into the fast proteins by adding peptide or single chain antibody ligands onto one of the sides of the protein, and they're displayed on the outside. And so we're utilizing those kind of platforms for oncology applications. We haven't seen it, and I guess it comes with the flip side of the longer development time. We need to be able to make that protein, purify it, characterize it, and it just wasn't feasible in a vaccine standpoint to even think about that.
Excellent. What about biodistribution of the particles, right? So it's an intramuscular injection. Do you see this stay at the local site of injection? Do you see it drain to local lymph node? Does it go systemic?
Yeah, that's a good question. So we formulated, we optimized the formulation when we inject into the muscle. We get very robust and local expression. So we see very little amount of the nanoparticles leak out of the muscle into the draining lymph nodes, for instance, or go systemically. Obviously, if they do go systemic, typically the liver and spleen take them up, and we don't see much evidence at all of expression in the liver and spleen after an intramuscular injection.
Okay, excellent. Let's see. All right, so a question coming in here, asking if you can comment a little more on the oral delivery options and possible induction of immune tolerance to the delivered antigens.
Yeah, so again, it's very early days for us looking at an oral-based vaccine. What we do have is pretty exciting evidence that if we deliver in animal models a formulation through the oral route, we get actually quite robust expression of plasmid DNA throughout the GI tract and what looks like beyond the GI tract. So I think from an expression perspective, we can achieve that with SARS-CoV-2 epitopes. The real question, and I'll admit I'm an oncologist, not an immunologist, that it looks like it will deliver orally. We're looking at generating different kinds of immunity, so more mucosal immunity. And so it remains to be seen whether that will be effective against a pathogen like SARS-CoV-2. But we're excited about doing that. This is sort of our oral vaccine project is sort of following after our intramuscular injection project. But I think if we do see correlates of protection like induced neutralizing antibody or cell-based response systemically, I think we're going to get really excited about this.
Excellent. Thank you very much. Well, it looks like that is all of the questions we have coming in at this point. So I want to thank you, John, for sharing your work and wish you the best and hope that we see a successful CoV-2 vaccine coming out of Entos.
Wonderful. It was great to talk to you, Jason. Fantastic questions, and it's been a real pleasure to be a part of JLabs and be exposed to fantastic folks like you.
Excellent. Thank you very much.
Thank you.
Great. I'd like to thank our speakers for this very informative discussion today. And to our audience, you will be receiving the recording of this webinar in an email tomorrow. If you have any questions about JLabs, feel free to get in touch at the email on the screen. And as a reminder, we run a number of online programs throughout the year, so be sure to check them out. On behalf of JLabs and our presenters, thank you for joining us today and have a great rest of your day.